Transcription can be broken down into three parts: initiation, elongation, and termination. During initiation, an enzyme called helicase binds to DNA and unwinds it, creating an initiation bubble. This space grants another enzyme, called RNA polymerase, access to a single strand of the DNA molecule. One strand of DNA serves as the template strand for RNA synthesis. During elongation, RNA polymerase matches the reciprocal nucleotide bases along the template strand in the 5' 3' direction, substituting uracil for thymine, but maintaining the other three bases, guanine, adenine, and cytosine. The resulting product is an unprocessed strand of mRNA. Termination is the final step of transcription. Termination results in the release of a newly synthesized mRNA strand from the elongation complex. The newly synthesized mRNA strand is then modified through a process known as mRNA processing. Through a process called splicing, non-coding segments of DNA, also known as introns, are cut out of the mRNA strand, leaving behind the coding segments, also called exons, behind. A poly-A tail and a 5' cap of phosphates is then added to the remaining mRNA strand to reinforce defense against destructive enzymes in the cytoplasm. After the mRNA strand has been modified, it moves outside of the cell nucleus into the cytoplasm. Translation can be broken down into three parts: initiation, elongation, and termination. During initiation, the mRNA strand binds to the small ribosomal unit, which consists of ribosomal RNA. A codon is a sequence of 3 nucleotides, which form a unit of genetic code in a DNA or RNA molecule. An anticodon is the codon's counterpart. The large ribosomal subunit attaches to the small subunit such that the first codon is aligned at the P binding site. This codon is also known as the start codon because of its specific sequence, AUG, initiates translation. In the cytoplasm, a tRNA molecule binds to an enzyme known as amino-acyl tRNA synthase. It is here where an amino acid is attached to the tRNA molecule. A tRNA molecule carries an amino acid whose anticodon pairs with the mRNA start codon. The anticodon attaches to the start codon at the P site, initiating elongation. During elongation, another tRNA molecule carrying an amino acid binds to the A site. With the help of an enzyme called peptidyltransferase, a peptide bond forms between the two adjacent amino acids and is carried by the molecule at the A site. The other worn-out tRNA molecule is ejected from the ribosomal complex at the E site, making room for other tRNA molecules and their anticodons. As this process repeats, amino acids will continue to bind together, forming a polypeptide chain that will eventually form a protein. A stop codon signals for the termination of translation. Once the stop codon has been reached, elongation will stop. A release factor protein initiates separation of the newly formed polypeptide chain. Once the chain is released, the ribosomal subunits separate.